find it easier to hear on that.
Yes, ma'am.
Okay.
Now I'm starting, ma'am.
Okay.
Hello. Good morning, all. It gives me an immense pleasure to extend a very warm welcome to all of you present here on the third day of our course on plant systematics, classical to molecular approach. Thank you all for joining us for today's session. For this course, we have highly qualified and eminent speakers who possess vast knowledge and experience in the field of plant systematics. Today, we are privileged to have Professor R. Geeta as our eminent speaker. I, on behalf of Gargi fraternity, welcome you, ma'am. Thank you, ma'am, for accepting our invitation. I express my gratitude to our principal, ma'am, Dr. Pramila Kumar. She has been our guiding light and has always encouraged us. It's my pleasure to extend a very warm welcome to all my dear superannuity teachers, Dr. Usha Prasad ma'am, Dr. Shashi Tyagi ma'am, Dr. Kiran Prabha ma'am, Dr. Geeta Mathur ma'am. You always motivate us by sharing your expertise and knowledge. Now, I think due to some urgent meeting, we have our annual day on 28th July. Principal ma'am not unable to join us, but she has sent her good wishes for us. Thank you, ma'am, for your wishes. Now I invite our superannuated teacher, Dr. Kiran Prabha ma'am, to give valuable inputs to our participants and also welcome the participants. Thank you, ma'am. Ma'am, mute hai, ma'am, mute hai.
Hello, am I audible?
Yeah.
Yes, ma'am. Yes, ma'am.
Okay. A very good morning to all the participants of the workshop. I am Dr. Kiran Prabha, a superannuated teacher from Gargi College. So I extend my welcome to the eminent speaker of the day, Dr. Professor R. Geeta. First of all, I would like to take this opportunity to congratulate our principal ma'am, Dr. Pramila Kumar, for always being motivating and initiating this start of this course. I also much appreciate the lead taken by Dr. Renu Soni and the whole team of Botany Department, Gargi College, for organizing this virtual short-term course for undergraduate students of plant sciences and life sciences on the very relevant topic of plant systematics. In the last 2 days of this course, they have covered, the eminent speaker, Dr. Satish Kumar, has covered brilliantly the topic related to the historical development of taxonomy and the branches of science, as well as the following the classical approach. And this has led to the development of a detailed historical account of development of classification from Linnaeus artificial system of classification to the natural and phylogenetic system of Bentham and Hooker and Angren and Parental. Moving on from that, in the last few decades, all of us know that there has been a much advancement in the newer techniques in the molecular biology, particularly in the DNA profiling, chloroplast DNA. DNA markers have been used, and they have been the major thrust towards proposing the phylogenetic system of classification of angiosperm. We popularly know it by APJ, and we are witnessing the advancement made by APJ4. So we see that inference from the molecular data has contributed significantly in framing the modern phylogenetic system of classification in the phylogenetic reconstruction, which has dominated the plant systematics in the current phase. In the present time, all of us know the situation of biodiversity depletion. We raise our concern about depleting biodiversity, about endemic species, about the exploration of mega biodiversity hotspots. And we are also concerned about the high rate of extinction of endangered species. So in this context, I would like to highlight that the importance of training in the plant systematics is very well appreciated and understood. We find that the recent trend of research in plant systematics is in the direction of exploring floras of the new region, and identifying the plant species as well as finding the evolutionary relationship of the amongst the taxa. So here the research in the using molecular data have been very, very useful approach in this particular line. Uh, so having known the importance of molecular biology and the plant systematics in today's context and in understanding the evolutionary relationships of the organisms in general and the plants in particular, very important. So today's session, we are fortunate to have the eminent speaker and the eminent researcher, Professor R. Geeta with us. I welcome again, I extend my warm welcome to Professor Geeta to join this session and to educate all of us. Professor Geeta has vast research experience and proficiency in the field of molecular systematics. Professor Geeta, as I know from her biodata that she, she has focused her research in the field of evolutionary biology and molecular plant systematics. I remember that she used to hold many workshops in the Department of Botany, Delhi University, where she was holding the post of professor for many years and was training the many teachers of the DU department by holding the workshop and training them in the field of molecular systematics and construct, particularly the very famous and the popular thing which I remember was she used to train the teachers in constructing the phylogenetic trees. And many a time I also wish that I should go and learn from you, ma'am. So I extend again the very warm welcome in this particular virtual uh, short-term course. I'm confident that knowing this recent molecular approach in the systematics and being motivated by your work, the students will be learning much more as well as the faculty members will also be learning. I also appreciate my, college students, the life science students and the botany students who have shown very great enthusiasm and interest in joining this course under the leadership of Dr. Renu Soni. And I'm very sure that this particular learning in the modern fields of molecular systematics is going to make a very definite impression on these young minds. And it is going to influence the need of the hour, and they will be making a better choices and the better decisions in the further um, careers and the research priorities. And this is the need of the hour that we need more modern taxonomists, and the taxonomy has already got its status parallel to many other advanced sciences. So thanks for giving me this opportunity to share my view and welcome our eminent speaker, Professor R. Geeta. And we are all here to listen to your expertise and get enriched. Thank you and all the best, Dr. Renu.
Thank you. Thank you, ma'am, for coming and sharing your ideas and knowledge with us. Now I invite Adrita Das, President, Gargi Botanical Society, Taru, to introduce our eminent speaker, Professor Argita.
Thank you, ma'am. I, Advita Das, welcome you all to today's session, and I'd like to take this opportunity to introduce to you our respected speaker for today, Professor R. Geeta, who will be giving us an insight on the topic phylogenetic trees, molecular phylogenetics, and systematics. Ma'am had completed her undergraduate and master's from the University of Delhi and taught at Miranda House after her master's for a couple of years. She served as a scientist in the Indian Council of Agricultural Research for 10 years and completed her PhD research at the University of Arizona, USA in 1997. Mem has done her postdoctoral work at the University of California and started as an assistant professor at the State University of New York and continued there for 12 years in the Department of Ecology and Evolution, after which she came back to India and joined with the University of Delhi as a professor in the Department of Botany from 2009 to 2018 and has retired recently. Professor Geeta has taught courses in botany and evolutionary biology, and her research interest lies in comparative, evolutionary, and phylogenetic biology. Her papers and publications include morphological and molecular evolution, plant history, and she has conducted several courses and workshops on phylogenetic biology in several places like India, Brazil, Indonesia, USA. Besides this, Ma'am has served as a member of the Science Committee, Biogenesis, Future Earth, and is presently the editor-in-chief, Phytomorphology, International Society of Plant Morphologists, Delhi, India. Ma'am has also served on the editorial boards of American Journal of Botany, Botanical Society of America, USA, and Journal of Genetics, Indian Academy of Sciences, Bangalore, India. With this, I'd like to welcome Ma'am amidst us. Ma'am, it is a privilege to have you with us today. Thank you.
Uh, Thank you, Adrita, for the wonderful introduction. Now I invite our speaker, Professor R. Geeta, to conduct her session on the topic phylogenetic tree, molecular phylogenetics, and systematics. Thank you, ma'am. Now you can.
Okay. Thank you. I'll just share that screen. For some reason, it's not showing up. Good morning and welcome to all of you. Thank you for being here and showing your interest in the topic of systematics. So I'm going to be talking to you about phylogenetic trees and how you interpret them and give you a sense of what I think systematics is. And then you're going to be getting more detail of that. So you should think of this as a kind of an introductory talk. Now, before I delve into my topic, I just want to point out to you that I currently in my retirement, I'm part of this group of scientists who are doing different kinds of activities. But what I wanted to sort of direct you to was this, if you go to the website, which is up here, and I encourage you to do that, we have resources for scientists, for teachers and students. And we've recently put out some exercises in phylogenetics. So we have this thing on topic of virus evolution, and we also have some phylogenetic exercises. So you might find this useful both to understand how the different kinds of viruses, what viruses are and so on, but also to get a sense of how you look at trees and make them grow. So this is a quotation that everybody probably is familiar with, but may not always have a good sense of what it means. But specifically and especially in systematics, it certainly doesn't make sense except in the light of evolution. So what is the aspects of evolution that are we think about? We think about evolutionary process, which is what's going on in populations where you have genetics, variation, change. Then there's splitting of populations, which is speciation. And we also, on the other side, we have the result of evolution, which is evolutionary history. And here, what we mean by this in this context, although there's many aspects of evolutionary history that we might allude to later on, the history of speciation and variation, which includes what's happening, intraspecific variation, interspecific variation, is what is encompassed in this evolutionary history. And that is what we are interested in when we want to do systematics. So hopefully people are familiar with the concepts of natural selection and genetic drift, where you start with an orange population and there's some variation, and that variation somehow gets changed and the population now is blue flowers, whatever. And therefore, we can think about the processes that are going on within populations. And this builds up the history of the groups that we are talking about. And if there is speciation, so in this side, we had species that are exchanging genes as a single population. individuals that are exchanging genes, and at some point there's a split and there are now two lineages, which in this figure will show up like this. You have the population splitting, there are two lineages, there's a change in one of the lineages, and then there's another split and there's further change. So you go from an ancestral orange population to something that is blue. One of this resultant species remains blue, the other one changes into green, and then there's speciation and further variation. And so now instead of one species, we now have three species. So that is how biodiversity is generated. And that is why we want to make evolutionary trees. And just to draw your attention to this nice little drawing in one of Darwin's notebooks, where he is obviously thinking about what might happen. And so he has this little sketch in which he has these lineages going and splitting and so on. So the point I want to make here is that the branching process is very much a part of evolutionary process, which is what we meant by when we refer to as speciation. So speciation leads to biodiversity. And that is why we want to think about it in the context of taxonomy. So one thing that we're all familiar with, we sort of is dinned into our ears, heads and ears and everywhere, senses, is that biological diversity is, there's classification. Classification is on the basis of shared similarities and differences. So if we look at the set of species, we might put the frog and the squirrel together because, say, they are vertebrates, they are vertebrate animals. Similarly, we might put the mango and the Dryopteris, the fern, together because they are vascular plants and they are plants and all of that. Okay, now what's the connection between these two different groups? Well, the fact is they are eukaryotes. And so we put them in a single group called the eukaryotes. And then we have the bacteria on this side. And so we sort of had this idea of eukaryotes and the bacteria. So that's a system of classification. And the reason why, and of course, everything belongs to life in general. They're all living beings. Now, this is called a nested hierarchy. So we have the lowest hierarchy. You might have species. There are many species within this group that might make up a genus or a family and so on. And here we have this group that we might call the vertebrates and so on. So this is a nested hierarchy in which we have the different groups which are sitting one inside the other. So the frog is a vertebrate. It's a frog. It's also a vertebrate. It's also a eukaryote, and it's also a living creature. So that's a nested hierarchy. So how does this nested hierarchy come about? And that exactly was the insight of many evolutionary biologists, including Darwin, who sort of realized that biological diversity almost invariably shows this nested hierarchy. And that, Darwin pointed out, is because of this kind of common ancestry. So we had a common ancestor that the frog and the squirrel shared. We had a common ancestor that the mango and Dryopteris shared. And there's a common ancestor that these whole groups share, which was a eukaryote, and so on until we have all of life. So the nested hierarchy is a direct consequence of this branching process of evolution. So nested hierarchy is explained by the branching process, and therefore we can think of the tree as a model of evolutionary processes. It explains the success of classical taxonomy. Here we had classically, we've talked about vertebrates, we have vascular plants, and within each we have a system of classification which more or less holds up quite a bit. Even today, even though there's been this advent, or I should say invasion, of molecular methods in phylogenetic systematics, but still our classical taxonomy has held up very well in many instances. And therefore, because we classify things according to this nested hierarchy, we are able to actually recapture, or in some sense, understand the evolutionary relationships. Now, in today's presentation, what I'd like to talk about is phylogenetic trees. We've sort of justified why we do phylogenetic trees. So I'd like to talk about them, how you read and interpret them, a very brief foray into maximum parsimony and how you reconstruct trees. This may be more than you wanted, but I thought it's good to give you a flavor of it. And what you mean, what one might mean by molecular phylogenetics or phylogenetic systematics. So let's just jump into the first step of reading trees. Now, why do I present this instead of how you get the trees first? Because I think more people are interested in the results of this, and more people and more generally can make sense of the biology they are interested in by looking at these trees. Not everybody is necessarily interested in building trees, although as a systematist you probably will be, but reading trees is something that is also not intuitive. So one needs to kind of understand how to interpret the results that you would get. So I prefer to do that first. So in this context, here we have a tree which consists of a branch, many branches. This branch is a tip or a leaf or an OTU. That's a term you may be familiar with. This branch is an internal branch. It's also called an internode because at the point in the tree, which consists of branches, we also have nodes and internodes. So between the two nodes is the internode, which we may call an internal branch. And then there's a special branch here, which is called the root. This is a rooted tree. And this is just a graph. Now a graph has to be interpreted. So in this tree, we are interpreting this graph, this branch as a species. This branch is a lineage of species. At the node is speciation. So that's what is going on in this very simple graph, but it's packed with information because remember, there's all the intraspecific processes of selection and genetic drift and migration and all of that going on in each of these sticks. But we're not going to be talking about it. But do keep in mind that this is not just a diagram that we've drawn for fun. So what does the tree depict? Certainly, it depicts a statement of relationships, right? We are saying that B is more closely related to C than it is to A because B and C share a recent common ancestor. They emerge from that ancestor, which specifically consists of this internode and this node. At the node, there was a speciation event. So this history is a statement of ancestry. It's also a history of speciation. So these are some of the basic things that a tree depicts. And again, it's useful, important, and necessary to keep these processes that underlie this figure that we might like to draw. Now, and it's through through the use of various kinds of data, molecular and morphological data, that we might then come up with this very, very abbreviated tree of life. I'm sure you've seen many more complicated pictures of the tree of life, but this is a tree of life, although we don't have many representatives here in these lineages. But given these taxa, this is how they're related one to the other. The thing to remember is that the tree is a strange kind of a graph in which really there is no scale. There is no scale this way, there is no scale this way. The maximum you can say is that these tips may represent the present and this root represents the past. So the scale, if it is, we can call it a scale, is that you're going from the present to the past, or if you prefer, go from the past to the present. So that's the only relative thing that you can say here. So the way to read a tree, regardless of whether it's drawn like this or like this or like this is how mathematicians like to draw it. You have to read the tree from the tip to the node, right? So we start with these two taxa, human and chimps, who share a most recent common ancestor, and we name that ancestor 1. And we say that these two are sister taxa because of this connection, the close connection between them. Now remember, we've not drawn all those Homo species that are out here along this lineage, nor the other chimp species, nor the other taxa that are along that lineage. Okay, so the human chimp lineage, so this is the lineage, is most closely related to the gorilla. They share the more recent common ancestor, that is number 2. And so on unto the relationship with orangutan, with which they share an ancestor 3. Now, I'm going to give you a little quiz. I can't see your responses, so I'll just give you half a minute to look at this tree and say whether or not it's the same tree as the first one. I hope many of you would have got the idea that the way, if you're reading it the way I said, read it from the tips to the nodes and not from left to right or right to left, you should have come to the conclusion that the chimp and the human in this tree are related one to the other. The gorilla, chimp, and human are related one to the other by ancestor 2, and so and with the orangutan through ancestor 3. So it's the exact same tree, because the chimp and human are sister taxa here, and their sister taxa here. The chimp-human lineage is sister to the gorilla, and this lineage, gorilla, chimp, and human, is sister to the orangutan. Now, each of these relationships, the sets of taxa which are held or which are commonly related because of sharing a recent common ancestor, is called a clade. So the chimp and human form a clade. Now they form what's called a monophyletic group, and the same, it's also called a clade. A clade simply means branch. In the context of phylogenetics, the branch is a monophyletic group. So there is no such thing as a monophyletic clade, or rather, don't use that term, because the clade is itself referring to a monophyletic group. Similarly, this group is monophyletic, that's a clade, and this is monophyletic, that's a clade. Now, the thing that jumps at you is that they are nested one within the other, which is what we referred to earlier as the nested system of hierarchy. So look at this tree. Is it the same tree? Well, I'm sure some of you have gotten confused because everything looks very different. But in fact, if you go from tip to nodes, you'll find that the human chimp are sisters with sharing common ancestor 1. They share the common ancestor with gorilla, that is the ancestor 2, and with the orangutan, that is ancestor 3. It's exact same tree, no difference. How about this one? I guess you now have gotten used to the idea that these are all the same tree, just drawn in different ways. So think of this like if you're familiar with the suspended thing called I'm forgetting the name. Well, I've forgotten the name of it. The thing that hangs, like hanging things, and they are related one to the other in the same way. Decorative structures. You'll realize that regardless how they are twisted around, it doesn't matter. They are still connected by the same point in that structure. So if we add to this tree the gibbon, okay, with which this set of organisms share a recent common ancestor, we find that this ancestor, this common ancestor, is what is called an ape, because the gibbons, chimps, gorillas, orangutans are apes. Okay, now this is a monophyletic group called the ape. Since we sit in that monophyletic group called the ape, what does that make us? An ape. So the answer to people who ask whether we descended from apes is no. We did not descend from apes, we are apes. And that's the way you should think about it. We are at the same time, we are Homo sapiens, we are Homo, the genus Homo, and so on until we are also apes, we are mammals, we are vertebrates, we are eukaryotes, we are animals, we are eukaryotes, and we are life, we are living things. So that's how the tree helps you to understand why we say that in systematics, we want to identify monophyletic groups. Because ever since Darwin, the attempt has been to try to have our taxonomy reflect evolution. So we'll be coming to that again, but there's just a couple more things about reading trees, in which you need to know about what a polytomy is, in which the tree relationships between the species A, B, C is uncertain. And the fact that these relations could be one of 3 types of relationships, maybe you can work it out. So what are the three possible? This is called the resolution of that polytomy, is that B may be most closely related to C, B and C are sisters, and A is outside. Or it may be that A and C are sisters and B is less closely related. Or it may be that A and B are sisters and C is the outside. So this is something to think about when you look at a polytomous tree. Now I find it very difficult because in lectures I usually ask, are there any questions? So maybe what you can do is to send your questions in the chat thing, and when we come to it, we will try to address those. Okay. So when we reconstruct trees, and so now I've given you a very brief overview of how the main things to keep in mind when you look at trees. There's many, many more things to keep in mind, which hopefully you learn as you go along. But let's have a look now at how we go about actually making these trees. The first thing is we are going to use some kind of characters, right? And the characters have to be homologous. Every systematics enterprise, every taxonomic enterprise has to start with characterizing species, comparing one species to the other by comparing homologous traits, and then doing what we want with it. So first thing we need to understand is homology. We're not going to go into it, but given an evolutionary view of this, homology is a trait that two species share with an ancestor who had that same trait. So they inherited it from that ancestor. We are mammals, we have hair on our bodies because the mammalian ancestor had hair on its body and we all have hair. So plants, the vascular plants, all have vascular tissue. Why? Because the vascular plant ancestor had vascular tissue, and that trait of having vascular tissue is homologous across all the diverse plants. Okay, so in the case of homology, then we can think about homology in the morphological context, which it seems to be increasingly students are not that familiar with because we seem to have molecular eyes rather than morphological eyes. But in this case, let's look at the seed. Okay, the seed in this zamia is homologous to the seed in this Elysium, which is a angiosperm. So we might choose a set of traits which are homologous one to the other, and then we might build up a data matrix, a taxon by character data matrix. So in this matrix, what we have are 5 taxa: Funaria, Equisetum, Dryopteris, Netum, and Mangifera. And we have 3 characters. We looked at the seed. The seed is a trait that is shared by Netum and Mangifera. So it's homologous in these, but the others don't have that character. In the same way, vascular tissue is shared by these taxa, but it's absent in the Funaria. Now, all of these are similar in that they have embryos, and that is a trait that they share exclusive of other non-land plants. So all land plants have embryos. Now, the same idea of homology is exactly applied in the case of molecules when we do an alignment. Okay, so what are we doing? First, we identify a homologous pair of homologous genes. We extract gene from this plant and from that plant, and we say, okay, these two represent the same genes on whatever set of criteria. I'm sure you're familiar with that. And then we try to line them up. We want to homologize, if you want to use that word, sites one to the other. So in this overall molecule, what we are trying to compare are the different sites where they may or may not have been a mutation. So therefore, when you do this whole exercise, we come up with an alignment in which we have to insert gaps, some 4 gaps in different places in order to maximize the matches between sites. Okay, so this whole exercise in the case of the seed has been done by generations of botanists, which you must have had a flavor of in yesterday's talk, yesterday and day before's talk, where we don't have to do that much hard work in the large context, but when you're studying a set of species of a particular genus, you have to do this work to figure out what's homologous with what. In the same way, you sequence of 2 genes, after that, it's not just automatic that you can do what you want. You have to put in this effort of alignment and then coming up with this data matrix. So here is a data matrix, exactly the same thing. Okay, so what I'm trying to drive here is that molecular systematics is not something magically different from whatever it is that we've been doing for centuries. What is different is the source of data and definitely some methods of analysis, which we'll again revisit at a point. Okay, so that's all I want to say about characters. Now about how we use these characters to classify, right? Classically, we have used overall similarity among species to say, okay, these two species have so many characters in common, therefore they are more, we put them in the same genus, whereas the other species looks different, it does not share as many traits in common, so we put it in a different genus. Now, this has been the approach starting from the classical approach, starting from Linnaeus and up to Cronquist and later. Of course, it didn't start with Linnaeus, but formally. It's also the approach in the field that we call numerical taxonomy. Now, numerical taxonomy is a sort of an interesting field that grew out of the attempt to try to make objective decisions about homology and character states. And so that is one stream that was coming through history. And this first was applied in bacteria. But so there the method of phonetics, the phonetic approach, and especially the UPGMA analysis is what is applied to come up with the way, or to come up with the phylogenetic tree. So the change that happens between over long time, between Linnaeus and the use of trees is been a very, very slow one. Initially, there was attempts to do evolutionary classification. And then with the addition of numerical taxonomy, the idea that you can make trees to reflect relationships. Although I must say that it was Darwin that introduced the idea, or brought into systematics and biology the idea of phylogenetic trees, or trees as showing relationships. So it's not so alien, but in taxonomy, it was something that Haeckel did, and then it went out of favor and then came back in the 1960s. Okay, so let's just revisit. Let's look at what do we mean when we say we are classifying based on similarities. Overall similarity. So here are the taxa we may call eubacteria, archaea, and eukarya. And here are different hypothetical characters, which I made up because it's just easier to do that. So these are 5 characters, A to E, and this is how the states look like. So eubacteria have some state, archaea has the same state, eukarya look different. So if you try to use a criterion of overall similarity, we'll see that there are 4 characters in which the bacteria and archaea are the same. And so we say these are more closely related to each other than either is to the eukarya, right? So overall similarity. Now, this has been, like I said, the way in which we have done classification for a long time. But the change that came in the 1960s was the attention, and actually it started in the 1950s, but it was in 60s that a person called Hennig kind of drew attention to the fact that there are some similarities between taxa, maybe due to different reasons, and that what we need to emphasize in classification is evolutionary similarity. And that led to this whole field of phylogenetics. So there are different kinds of homology, was his point. He says there are some kinds of homology which is ancestrally present. So here, let's say we know we are God, and we know these species are related like this with each other, and they retain the similarity of this ancestral state, right? And this ancestral state is something that is retained in the species A and in species B, and in this result of speciation, which led to this lineage, until at some point, red became blue. Okay, that evolutionary change is an derived similarity. Okay, and that's what has been given the names plesiomorphy and apomorphy. Normally, I don't like to use terms, but I know that it's probably there in various syllabuses. And it was Hennig that introduced these terms. So the retained ancestral similarity is a plesiomorphy, and the derived similarity is the apomorphy. When we say derived, we mean that in the history of these species, these two species have this trait because they inherited it from that ancestor within which the change took place, whether due to natural selection or genetic drift or anything, we don't know. And at a certain level, we don't really worry too much about it. So the similarity between A and B in this case is due to this retained similarity, or it's due to the simplesiomorphy, shared retained similarities. On the other hand, the similarity between C and D is due to shared derived similarity, or it's a synapomorphy. So Henig's insight was that monophyletic groups should be recognized by these unique synapomorphies, and that this is the only homology which points to historical relationships in this particular group. Now, if this particular group had another species E in it, and that E had a green trait for the same homologous trait, then we would say that here the change happened here, and then there was another change. Okay, but within, and within the set of species, it is this trait, the blue trait, that is indicative of relationship, and that was the field that was then called cladistics. Meyer actually gave it that name. Okay, so if we try to do the same, revisit this data matrix using this approach and say that, okay, in this case, we know, because we are God, that 0 is the state that was ancestrally present when life arose, and 1 is the derived state. Okay, so when we look at this data matrix, how many derived character states are there in this tree? There's only 1, and that is shared by archaea and eukarya. So if we use the Henigian principle that you should only use shared derived characters as indicative of relationship, then this is what we would get. We would put the archaea and eukarya together, and we would make a group, and that trait is characterizing archaea and eukarya. Now, why have I drawn these branches of different lengths? Because the rate of evolution in this set of characters is much higher in eukarya, and so it has a much longer branch length. And that was what was misleading us, because in these characters, eubacteria and archaea did not change much. Whereas these, the eukarya, after divergence from the common ancestor shared with archaea, has changed a lot. So that in principle is the difference between the use of overall similarity and evolutionary similarities in trying to apply in classification. So we will kind of do that using parsimony. And we'll revisit the character matrix that we had built up earlier on morphological characters. So if we take the character seed, and we realize that it's only Netum and Mangifera that share this trait, we say that this shared derived trait unites these two into one group. Okay, and we put them together. Then we take the vascular tissue character, and we say that Equisetum, Dryopteris, Netum, and Mangifera share that trait, and we put them all in one. Now, note that there is a polytomy here. We don't know how Equisetum and Dryopteris are related one to the other. Is Dryopteris maybe more closely related to the seed plants? Or is Equisetum maybe more closely related to the seed plants? We can't tell here, because our character matrix does not contain information on that score. What it does tell us is that all of these taxa have embryos, and we know from other information that this embryo was in the ancestor of all these land plants, and therefore there was no change at the root of this tree, but it happened in a previous, in a deeper in the tree. So this is how we come up with this tree. It is not fully resolved. Why? Because there's this one polytomy where we are not able to tell the relationships within the vascular plants, but we are sure that the seed plants form a group. We are sure that the vascular plants form a group. Okay. Now let's do the same thing with that molecular data matrix that we had. Now remember, we are only going to take into consideration the shared derived similarities. And when we look through the data, through the alignment, we see that there are 5 characters. Okay, and I'm not going to go through this step by step because it probably take too long. But believe me when I say that it's molecular data 1, characters 1 and 3, which unite this group here. And here we have something which we did not have in the morphological data, which is molecular trait 2, which actually serves to put Equisetum and Dryopteris together. And then we have 4 and 5, which separate the vascular plants as a separate group. Okay, so we've added some resolution to the previous tree, which however was very similar to the one we have now. No big surprises there, but it has helped us to resolve this polytomy where we now can say that Equisystem and Raptorus are sister taxa without any doubt. Is that true that it is without any doubt? Well, we'll have to visit that. But this is a exercise that we do using, it's easy to do when you have 5 characters or, 5 taxa, in this case, maybe some 20 characters. It's painfully slow. But it's, manually doing it is a little difficult. So the parsimony analysis actually allows you to do this manually, also again with a smaller data set, but I want to sort of introduce you to the basic principles of parsimony analysis so that you can have an idea of how it is done when a computer does it. So your grouping taxa based on shared derived character states, that is synapomorphies. And if there are many possible trees, what you're going to do is pick the one that is the shortest tree, which shows the fewest changes, the most parsimonious tree. Okay, so here's a data matrix, anonymous taxa and anonymous characters. And what we're going to do is look at what each character is telling us. So here we're saying B and C, okay, go together according to character 1. Character 2, what does it say? Yes, it says A and B go together. What does character 3 say? Does it say a different thing? No, it says the same thing as character 1. So we have a case here that all the characters are not saying the same thing. In the data matrix that I showed you earlier, they're all saying the same thing. They're not contradicting each other. They're consistent with one another. But real character data sets are going to be messy like this. Some characters will say something, others will say another thing. So parsimony analysis, what it does is here, so different characters are saying suggest different trees. So which tree should we pick? We use the second criterion, which is that we pick the tree with fewest changes. So there are 2 possible trees in this case, right? Where B and C are going together and A and B are going together. We have to choose between the 2. So we will pick the one that has the fewest changes. But now so what we're going to ask is how many changes are required in all the characters, and that's what a tree length is. How many changes are required by each tree? And that's what we will do as an exercise. I'm not sure I have enough time for this, but we'll try. Okay, so what I'm going to do is to present you with 3 trees, even though our character matrix actually only supports 2 trees, just so you'll know how the computer does it. The computer will say, okay, if there are 3 taxa, here are the 3 possible rooted trees. Here's my character matrix. I'm going to see how each character set fits on this tree, how many steps are required by this tree, and how many steps are required by the 3rd tree. Okay, so character 1 is unites B and C. It's in the common ancestor of B and C. What about character 2? Because we know the ancestral state is 0, we know this. We can ask questions about that later, but here we know it. We say that character 2 evolves 2 times, once in A and once in B. How about character 3? Again, it evolves just once in the common ancestor of B and C. So what we see is that we have to count the number of steps taken by this tree, required by this tree to explain the character matrix, and that is 4 steps. If we do the same exercise with the second tree, A and B are together on this tree, and so character 1 has to have evolved 2 times on this tree. Character 2 evolves just once, it unites A and B. Character 3 is again like character 1, it evolves 2 times. Now this may go a little fast for you, but I want to give you the flavor, and I will try to share some of these things at a later point. Okay, so how about, and that takes 5 steps. So if we look at character, the 3rd tree, can you guess whether it's going to be shorter than in either of these trees? Okay, I don't know what you said, but B and C are far away. I mean, they're not so closely related to each other. So the character 1 evolved 2 times, once in B, once in C. Character 2 evolves 2 times, once in A, once in B. So you see where we are going. And character 3 evolves 2 times, once in B and once in C. So the total number of steps is 6. Therefore, by the criterion of parsimony, maximum parsimony, we choose character tree as being a character, I'm sorry, the first tree, tree 1, as being the one that explains the best way the data here. So there is another thing that you might like to know about, which is how do we know whether these results are robust or not? And I think I'm not going to go through the exercise, but just to tell you that it's a way of pseudo replication where, because here it's difficult to replicate this experiment to know how well we can, how confident we can be of our result, what we do is something called a pseudo replication. And in this particular run, here is our original character matrix. We have resampled with replacement 3 times. And this is our bootstrap analysis. In this one, we see that A and B go together according to this character matrix. Here, B and C go together. Here, B and C go together. So the bootstrap support is that of all the bootstrap analyses done, 2 out of 3 results have shown the same result as our original tree. And so we say the bootstrap support for B and C being sisters is 67%. So I realize that I rushed through this. More characters, we might get a result like this, where some branches have more than 95% support or 90% support, and some have less than 90% support. Okay, and those I've colored pink. So this is what we get, but we don't believe all those branches. What we should believe in is that we collapse all those branches and make polytomials. So we don't believe this branch that separates taxon 1 and 2, and we say 1, 2, and 3 make a polytomy, and so on. Again, I don't want to dwell in this, but this is what bootstrap support analysis tells you. So in this case, in the different tree now, we have a polytomous tree, but we can be more confident. We have more confidence in this tree than in the tree on the left. So these are the ways in which, because this is a scientific process, we are not imagining this, we are not making it up. We are using data to support hypotheses. Trees are hypotheses. So the support for them may be, the support for our hypothesis may be strong or weak. Now, in this case, some parts of our hypothesis is weak, right? Therefore, if we add more taxa or we add more characters, we may get better support for the hypothesis, or it may be that this hypothesis, the original hypothesis may get blown and we may have something else. So this is how we get uncertainties in phylogenetic analysis, why things change over time. So I just want to spend a little time talking about, okay, in this case, we did molecular and phylogenetic analysis, morphological and molecular phylogenetic analysis. Was there any difference? I don't think so. So in principle, they are exactly the same thing. What data are you using? How are you analyzing it? That is the question. That's what you have to see whether there's a difference or not. So molecular phylogenetic analysis is not different from morphological phylogenetic analysis. Then why is there so much attention on molecular phylogenetics or molecular systematics, right? Well, one thing is that there's lots more data. Sequences are long. If the variability is good enough, then you get many more characters than you ever can in a morphological analysis. And therefore, the robustness of your results might be better. Now, that's a general statement. Sometimes it's true, sometimes it's not. Okay, so a lot of insights that we are now getting in molecular systematics is the addition of data, something like the example I gave you earlier, where molecular data gave you the idea, the notion that equisetum and Dryopteris may be the closest relatives. Okay, so you can have additional information and a shoring up or a support, stronger support for original existing. Now, in taxonomy, what can you do with molecular data? And we've talked about building trees, but I want to revisit one more thing on which I think tomorrow Dr. Stalin will be talking to you about. So one is identifying and naming, which is an intrinsic part of taxonomy, right? What we've talked about so far is only the classification part of taxonomy. So how can molecular data be useful there? And that's what we want to know is what is this entity we have in hand? We want to identify it. We want to identify it by using DNA barcoding, or that is what I would rightly call molecular taxonomy. Okay, the taxonomy has to do in this case with identifying and naming and then placing it in a classification. Okay, that is the taxonomy. right? You can also identify fossils using phylogenetic methods, using molecular data, because some fossils will give you molecular data. So the whole history of human evolution is based on this kind of analysis. Now, when you classify, what we want to ask is not what is its name or what group does it belong to, as much as how are these taxa or genes related to each other. So we are classifying the taxa, we may be classifying the proteins, we want to identify whether some gene copies in two different species are homologous or paralogous and also on. So I'm not going to go into all of that. But I just want to end this section by just saying that we want to know, it is essential and it's useful to know how organisms are related. I hope we've sort of conveyed that in the context of taxonomy, that molecular data can certainly be useful in this context, but there are many other fields of biology that, you know, this information is useful. And now one thing that you can't avoid in this context is epidemiology, right? We have phylogenetic trees everywhere. They're reporting about where did the Wuhan virus come from? Was it Chinese? Was it genetically engineered? No, it was not. So all those kinds of questions are answered partly, at least, using phylogenetic methods. Okay, now let us then look at what we have just talked about. Molecular systematics and morphological systematics then are actually, you could call them together as just phylogenetic systematics, which is what I prefer. I don't like to talk about molecular and morphological systematics as if they are two different things. And they essentially are about classification. We are putting them in a system. Okay, that's what Hennig first called it. Systematization, putting them in a tree, seeing who's more closely related to whom by determining who is their, whom they share ancestors with. So that is what phylogenetic systematics is. So now we'll just go into phylogenetic systematics and talk about a few ways in which phylogenetic systematics is applied or can be applied. Can be done using phylogenetic trees in the taxonomic context. Okay, and here then, let me also just say one word about molecular and morphological systematics. It is not so clear to everyone, and everybody does not agree about how to deal with morphological data and molecular data together. Okay, so there is some amount of disagreement. Most people would say that molecular data are much more informative, they're more reliable, they trace the genetics of the organisms, whereas morphology is a phenotypic trait. We can't be sure about the genetics underlying that phenotypic trait, and therefore you should only use genetic data. Okay. Others would say that the morphology is equally a part of the history of the organisms that you're looking at, and therefore you have to put it in the context of the molecular data to determine what it is. Okay. Now, the people fall short of killing each other on this question, but there's a lot of contention about it. And usually you have, just like everything else in this world today, you stick to one or the other point. And you have to take the kind of decision as to whether you can evaluate everything. And my own view on this is that if everything agrees, then that's the result I'm going to agree with. And the rest of it might or might not be debatable. Okay. So when we look at phylogenetic systematics, what is the goal of phylogenetic systematics? I alluded to that earlier, but now we're going to deal with it more explicitly. Primarily, it is to classify organisms according to their evolutionary relationships. Okay. Now, already this takes out the barcoding business out of it. Okay, because barcoding is about molecular taxonomy for the identification and putting it into that context of known species. Right? So classifying organisms according to their evolutionary relationships is the main aim of phylogenetic systematics. And further, what do we mean by evolutionary relationships? We want to recognize and name only clades. We only want to recognize clades and give them names. Okay, if something is not a clade, we don't want to use a name for it. Okay, so taxonomic names should only be applied to clades or monophyletic groups. So that is, it's as simple as that. And that is the basic reason why there's so much grief. All right, let's just visit something in which people will kind of relate to more readily. So let's now look at this tree, phylogenetic tree, which is different from the one we looked at earlier. But let's say we are botanists and we are looking at plants, and we have the bryophytes and pteridophytes put together. They share a most recent common ancestor. And that ancestor, remember, is this ancestor, and it's the branch subtending it in this tree. Okay, we can also look at the fungi who have that share that common ancestor, and which then put all the ascomycetes, basidiomycetes, and a few other groups together. But remember that oomycetes and some of the other fungal groups are outside. Okay, we have the amniotes, the egg-bearing birds and mammals, which share a most recent common ancestor. We have the vertebrates, which include the amphibia and birds and mammals. And we have animals, which include things like butterflies. But we also note that the fungi share a more recent common ancestor with animals. So I always like to say that my mother is right. She doesn't eat mushrooms because she's a strict vegetarian. So mushrooms are actually more closely related to animals than they are to plants. But I hope I don't reconvert some of you into non-mushroom eating creatures, because that might be carrying it a little too far, but it's an interesting fact. Okay. So a monophyletic group or a clade includes an ancestral taxon and all of its descendants. Okay. So here is a vertebrate ancestor and all of its descendants, amphibia, birds, and mammals. Here is a land plant ancestor and all of its descendants, in this case, including bryophytes and pteridophytes. Okay, now, if you look at the same tree, here is a group, right? Is it a monophyletic group? So think of what ancestor is shared by this monophyletic group, if it were monophyletic. This group has a common ancestor in this blue circle that's written down here, right? Now, the descendants of this ancestor include not only bryophytes, pteridophytes, and fungi, but also insects, amphibia, bird, aves, and mammals, right? So, if a monophyletic group is one, what was the definition? An ancestor and all of its descendants, then we also are plants if we want to call fungi groups. So these are the kind of counterintuitive results that come from phylogenetic systematics and which people don't like. We want to continue. I think now the resistance against fungi being taken out of plants is probably gone, but still botany departments are the ones that teach fungi. So to go figure. So a non-monophyletic group includes an ancestral species and some, but not all of its descendants. So in this case, we have this ancestral species, and the plants as used to be defined, including fungi, would include bryophytes, pteridophytes, fungi, and all the other plant taxa, but not the animal taxa. So that is a non-monophyletic group. Sister taxa are each other's closest relatives. In this case, sister taxa can be identified. We have the aves and mammalia that are sister taxa. We have bryophytes and pteridophytes that are sister taxa. Plants defined as plants, bryophytes and pteridophytes, which are sister to the fungi and animal taxa. So these are the ways in which we interpret this tree. Okay. Now, non-monophyletic groups may be said to be paraphyletic. That is, these lineages resemble each other but are not sister taxa. So when we revisit this example, these are similar only in that they are sedentary organisms with cell walls. So that is the one thing in which fungi resemble plants. In most other traits, they are different. So paraphyletic groups resemble each other but are not sister taxa. Okay. Now, here is another group that we've already met, the so-called prokaryotes. We know today that archaea are more closely related to the eukarya, but historically, prokaryotes are a group that we have in the past recognized, which included the bacteria and the archaea. So that would be prokaryotes would be a paraphyletic. So which taxa are sisters in this case? it would be the archaea and eukarya. Okay. So many non-monophyletic groups are, many paraphyletic groups have been recognized by us in the past. For instance, pteridophytes. We know mainly because of molecular data, but there's a lot of support from the morphological data, that what we used to call pteridophytes, Selaginella, the lineage that's now called the lycopods, the lycopod lilies, and the ferns and so on, are actually in two different lineages, right? So Selaginella, this ancestral species, speciated, gave rise to the Selaginella line and to this other line, which includes Equisetum dryopteris and seed plants. So the pteridophytes are not monophyletic. Now, this has also been the case, it's been also considered the case for bryophytes, where the liverworts and hornworts and mosses are not necessarily monophyletic. And this has been, but it's been kind of somewhat controversial. And recently, phylogenomics data seem to suggest that bryophytes are monophyletic. So we have to see how that works out in the future. All right. So there may be other taxa that are polyphyletic. So if we look at this, what happened to plants? What happened to algae? Are they monophyletic? So if we look at this tree and look at just the branch that we call eukarya, right? And here is a expanded version of the eukarya, which we have ignored up until this point. The green names and lineages are what we used to call plants. So they include not only our, green algae and green plants, land plants, and so on. They include the red algae and the closest relatives. They include the euglena, which of course the zoologists have always claimed, and we botanists have always claimed, and other lineages like the Cryptophyta, the Haptophyta, Alveolata, Staminophylla include things like familiar things like diatoms and brown algae. So all of these taxa we used to call plants. Why? Based on their photosynthetic nature and their sedentary nature and some other traits, right? Photosynthesis being the primary. But this is a non-monophyletic group, which is polyphyletic, because it is difficult to know, it's just comes up in different places on the tree. So let's then look at what happened to angiosperms, right? So this is what you're going to spend some time in the coming sessions on looking at. And the angiosperms are still monophyletic, right? So that's a good thing. What happened to dicots? Well, the dicots are no longer monophyletic because you have a lineage called the eudicots, which may be more closely related to things like monocots. And in any case, these lineages, what used to be called monocots, what used to be called dicots, are now at the basal, so-called basal angiosperms, right? So these are the ways in which our classification system have been modified a great deal. But you still, within the eudicots, have something that you can call the rosids. You still have the asterids. And it is the, the APG classification is trying to clean up some of the messes. But I would say that in many cases, the kind of, if there was uncertainty in morphological data, very often there continues to be uncertainty in molecular data. And therefore, you should not always expect that molecular data will solve the problem that you think you are trying to answer. Now, for instance, let's just go back to that bryophyte example, right? For a long time, we thought that they were monophyletic. And then there was a period of, I would say, 20, 30, 40 years when that was brought into doubt because of the increasing use of molecular data in analyzing these taxa and trying to get phylogenetic relationships. And now in the past couple of years, actually, this result that I've put here with phylogenomic data comes up in the last, within the last 6 months, although there have been hints in previous analyses, and suggesting that they may actually be monophyletic. So, at every point, what's important is that you have to remember these are hypotheses, but hypotheses are not imaginations. They're not speculations based on anything else, on nothing, right? A hypothesis is a reasonable explanation of data that we have on hand. That's what a scientific process does. It tries to look at, here, I think that bryophytes are monophyletic. What do the data tell me? The older data don't support that very well. Maybe the newer data will continue to support it. Okay, so nothing is set in stone, and there is no absolute truth in any science, let alone in phylogenetic systematics. But as we go along and we use more and more data, we come to better and better understanding of the taxa that we are interested in. Okay, so what we've done in this looks like it's less than an hour, is that we've looked at phylogenetic trees, tried to justify the use of phylogenetic trees. I mean, it's not just a diagram. You don't draw the phylogenetic tree. Of course you do, but only once you have analyzed your data and said that this is the best explanation of the data I have on hand, whether it's morphological or molecular. We read how to read and interpret trees. We've looked at very briefly at how to reconstruct trees using maximum parsimony, and showing you that in principle, the same way is used for analyzing morphological and molecular data. But then there are many methods like the maximum likelihood method, the Bayesian methods, and neighbor joining methods, some of which are not applicable as yet to morphological data, and which can only be applied to molecular data, especially the maximum likelihood methods. And because the maximum likelihood and Bayesian methods are statistical methods, many people prefer those, because you can have a better understanding of the support for your the hypothesis than in the case of use of maximum parsimony. But in many cases, you have to, I like to introduce maximum parsimony because it's intuitively understandable. I hope that our little exercise, and if you didn't get it, I can revisit it later during question hour, should have given you a feeling for how we can use maximum parsimony in analyzing data, whether it is to build trees or whether it is to look at the evolution of particular trees. That's also possible. Now, we also looked at what is molecular phylogenetics and what molecular systematics is. And I suggested that molecular systematics is not anything different. Molecular phylogenetics is not anything different. And all of this is what is preferably called phylogenetic systematics. I realize that that's not how it's often referred to in many textbooks or otherwise, but this is my view of the field here. So if you have questions, of course, right now, feel free to, I'm sure you'll ask them, but eventually I'm happy to answer any questions. Just don't ask me, tell me all about this thing. You have to give an sense that you have actually read something, understood it, and it will be just one question. I can't give you another lecture in writing. I don't have that time, but I'm happy to answer specific questions by email. Now, before I stop, firstly, of course, I want to thank you all. Thank Renu for inviting me, and the Gitas. Nice to meet up with them again, the various Gitas, and all of you for attending. It's interesting. I can't say thank you for sitting patiently, because you might have been doing anything and I don't know. But anyway, thank you very much. And I just wanted to again remind you that in these strange times, and I don't know what's happening, whether you'll be able to go back to college or not, but whatever it is, don't forget that even if things open up, the dangers of contracting the virus are still there, and do remember all the precautions. Even now, going out to markets, I see especially young people, I'm sorry, and many of you are probably in that class. You are not impervious, and people like us are more susceptible because you think that you are not impervious. You might be carriers. So please take that care. All right. So if you have questions, I'm happy to answer. And I think at this point, I don't know what I have to do. Should I stop share?
Yes, ma'am. Thank you, Professor Geeta, for a highly comprehensive session. It was highly engaging, and right from the basics to the advanced concepts, you dealt with all the concepts very clearly. Now I invite Dr. Renu Puri to say a few words for ma'am. And I must tell you all that Dr. Renu had been associated with Gargi College as a faculty, and she also happened to be one of Professor Geeta's PhD scholars during ma'am's tenure in Delhi University. Dr. Renu, over to you. Are you there?
Nice surprise.
Thank you, Akansha. Thank you so much. And I would like to just add a few things about Professor Geeta. It was so lovely to work with her, and I'm privileged. I feel privileged that some of the students whom I've taught is learning from you, and I've been your greatest fan. As a student, still now you have the same enthusiasm. And as one of your book, when I was searching for some book in your library, one of your colleague wrote that you are a wonderful person, a person who's ready to share. And this is one quality I think every teacher should have. And today you have the same enthusiasm and that sharing capacity. So thank you so much, ma'am. It was very nice to see you again. And thank you all my colleagues. It's so nice to see you all again. Thank you. Thank you, ma'am.
Thank you, Renu. Now I would like to quickly just take up a few queries raised by our audience connected to us via YouTube's live streaming. I request Professor Geeta to quickly guide us all through these queries. The first question is from Nitesh Chauhan, and he asks, please explain nested hierarchy once again.
Okay, I think I'll go back to the presentation because I think it'll be simplest to do that. I'll just 1 minute. Is that okay?
Yes, ma'am.
So what we mean by nested hierarchy is just this. I just want to go back to this particular slide. Okay. So if you were to think of, and I'll go back to the original slide earlier, but if you think of just these clades as being, let's think of this as one family, chimp and human family. And then we're thinking of the gorilla, chimp and human something else. Call it an order if you like, because that's how we like to do it. Or we want to think of this whole thing, including, and if we include the gibbons also, maybe that's the best way to do it, then we are now calling this whole group the apes. Okay, that's the largest monophyletic group, which is the apes. And then we have the, within that, nested within it, is this group that contains the orang, gorilla, chimp, and human. Within that is nested this gorilla, chimp, human group, and within that the chimp, human group. So if we classify according to this criterion, I mean the classification that we come up with in the case of, here the nested hierarchy is these animals form a group. Within this group, the frogs, amphibians are nested within this group we call vertebrates. Squirrels are nested within this group called vertebrates. Squirrels are mammals. So we've not shown everything on this tree. So a tree, remember, is a simplification. Within this group that we are calling plants, we have the flowering plants, we have the ferns, and we have other plants as well, right? So those are nested one within the other. So the way to think about it is, in this case, let us say the mango is a flowering plant. It's also a vascular plant. It is also a plant, a land plant. It is also a, say, eukaryote, right? So the same, think of another kind of classification. What you have in the army. You have the Jawan and you have the general. The general does not include the Jawan, right? That's not a nested hierarchy. That's a hierarchy of a different kind. You have teachers and you have students. You have professors and you have students, right? That's also a different kind of a hierarchy, but it's not a nested hierarchy. The teachers don't include the students. The general does not include all the majors and whatever sequence of this thing there is. So that's like a ladder. In biology, the point is it's a tree. It's not a ladder. So our hierarchy is not a linear hierarchy. It is a tree-like structure. Therefore, we have a nested hierarchy. I hope that was clear. Otherwise, you can ask again, I suppose. Right?
Nitesh, are you here? Are you satisfied with the answer? All right, let's move to the next question then. This question is from Raghavendra Niranjan, and he asks, please differentiate between plesiomorphy and synapomorphy.
Okay, so again, I'm going to go back to the presentation. I have to remember where it is. Oh, it's after this. Okay.
Geeta ma'am,
Yeah.
could you please share your screen?
Yeah, I'm just going to do that. I'm trying to find the slide I'm looking for.
Yes,
Sorry.
ma'am.
Sorry, interest. All right, share screen. Maybe I should not. I can do it like this. I think this will be easier. I'll just leave it here. So here, I realize that there's a little bit of maybe the show maybe work better here. So what I'm saying here is that there is a trait which is there present ancestrally in this group. Okay. Now, within this group, some of the what's called basal lineages retain that trait, right? But because there are other taxa within the same group which have a different trait, this ancestral trait is not indicative of relationships. It is just something that is there in the group. It's sort of like saying, so let us think of there being a cat and a dog and a ape. Okay, you cannot say that the cat and dog are more closely related because they have hair, because the ape also has hair. But the naked ape, humans, supposedly lost hair. So if you want to say that, we can't say that the cats and human and dogs are more closely related to each other because they have more hair. Because that is an ancestral trait. That's a plesiomorphy. That's not indicative of relationships. What is indicative of relationships is what happened in history within this group, so that you can say, we know that, say, based on something else, we know that A, B, C, D are in one group. Let's say we know that land plants are in one group. What makes us say that ferns are more closely related to seed plants than to lycopods? Okay, there we don't have a good, well, we do have a good trait, and that is that they have what are called megaphylls. They have large leaves. Lycopods have simple leaves. They have microphylls. Right, that's only one trait. It's not, so it's something that we came after the fact and we said, oh yeah, look at this this molecular tree, and we can see that this megaphyll is something that actually unites, is something that evolved after separation from the lineage that the lineage shares, the common ancestor it shares with the other taxa. So I hope I'm not getting complicating this, but point is this trait comes later, right? And that is an apomorphy. So a synapomorphy is a shared derived similarity, and a plesiomorphy is a shared retained ancestral similarity. So when you say that a shared similarity that is plesiomorphic is not indicative of relationship, we should not use it as a homologous trait, because it's going to mislead us. Because it's going to tell us that A and B are more closely related to each other. Whereas in fact, because we want to use only evolutionary similarities as evidence of relationships, it's actually C and D in this case that are sharing, related to each other. Now, like we saw in our exercise on parsimony, characters can contradict each other. And then we have to make decisions. And sometimes our result will not be well supported. And we just have to live with that. We just have to live with polytomies. So anyway, so I hope that answered the question about plesiomorphy and apomorphy. Was that the question? Who was that who asked me?
Raghavendra Niranjan.
No, I meant you. So is that what the question was?
Akanksha. Yes, ma'am.
Yeah, Akanksha. Okay. Yeah.
Okay, let's move
Alright.
to the next question then. This question is asked by Prashant and Shivali Sharma. They want you to please re-explain pseudo replication.
I didn't explain it, so I can't really. I'll explain it now. I thought I'll run out or short of time, so I didn't do it. Okay. So now let's go back to the. So this was the original data set, right? This is what we built our maximum parsimony tree, saying that ABC, BC relationship was the most parsimonious based on the number of steps. Okay, so here we start with this tree and we want to know what's the support for this relationship. That is, in this tree, we are saying that B and C are more closely related to each other than either is to A. All right, so what's the support for that? So we say we do pseudo replication, which is, think of it like you have these columns of characters, okay? So we have this is original data set. It's only 3 characters, so we don't have much choice. But what we are assuming is that this set, this morphological data set or whatever data set is representative of the world of characters that these 3 taxa share. Okay, so you're putting it into a bag and you're blindly sampling. So you pull out character 1, all right? And you put it into your new character in your pseudo replicate. This is a pseudo replicate because you are not actually replicating the results. You can't, right? There's only one set of taxa, there's only one set of characters. You can't replicate it in that sense. You can't, even if you ran evolution again, you might get a different result. That's what Stephen Jay Gould said. Okay, so you can't replicate it. So the pseudo replication is what's called a bootstrap analysis. I mean, bootstrap analysis is a way of pseudo replication. So in this new character data set, you are putting this character 1 and you're actually now, in a sense, also putting it back. So it's sampling with replacement. So you do that multiple times and you come up with this new character set, the data set, which is the same size. So it's the same number of taxa and the same number of characters. So in this case, as you'll see, character 1 is picked 2 times. Okay. So character 2 doesn't have a presence at all in this pseudo replicate. So you build a tree based on that character matrix. And in this one, all characters say the same thing, that B and C are sisters, right? So all of them say that the state 1 is shared by B and C. So B and C are sisters. You do the same replication thing exercise for another character, for a different replicate, pseudo replicate. And there we find that character 2 is picked 2 times, okay? And character 3 only once. So this tree, not surprisingly, wants to say that A and B are closely related, are the sisters. Now the third replicate says that it picks character. Obviously, I've made it like this. So character 3 is picked twice, character 2 is picked once, character 1 is not picked at all. So here again, B and C are related one to the other because that's what the overwhelming, the maximum, think back to how the maximum parsimony tree would be, right? So there this will be the shortest tree. You'll have to go through the exercise again to kind of internalize that. Okay, so now you'll note, supposing there were 100 characters, okay, and most of them had this pattern of 0, 1, 1, that 0 in A, 1 in B, 1 in C. Then regardless of how many times you pick that thing, you're more likely to come up with the results that B and C are more closely related to each other than either is to A, right? That's because most of the characters say that. So the more the number of characters that say the same thing, the more robust will be your results. At the same time, you can't just ignore the ones that say it's something different. And that's why we have bootstrapping. Okay, so there are different ways of assessing. A bootstrap analysis, by the way, has the original, the word that the phrase that comes from is just bootstrapping is to, if you fall, there's no one to help you. You're pulling yourself up by your bootstraps. Okay, that's the thing that you're holding to pull yourself up. That's the bootstrap. So you're bootstrapping it, you're using your own efforts to pull yourself up. So in this case, we're using the same data that we have to try to get some sense of how robust the results are. Okay. Anything more? Do you want me to go over that other tree also, or just the analysis?
I think that's fine, ma'am.
It's okay, right? Yeah, okay.
Okay, let's move ahead. Next question, rather a couple of questions are from Divya, and she asks, what is the use of bootstrap value in a phylogenetic tree, and what is the interpretation that we can do if bootstrap values are given in a phylogenetic tree?
Yeah.
And the next question is also related to this one. When we make a phylogenetic tree, we have bootstrap values like 500, 1000, etc. What different does it make?
Okay. So the 500,000, etc., is something that will depend upon how many bootstrap replicates you run. In the fake bootstrap replicate that I ran here, I only ran 3 replicates, which is no good. Okay, but you can't do that manually very easily. So in bootstrap analysis nowadays, and early days when I started this business, which was in the dark ages, bootstrap runs of 100 was thought to be very good. Okay, now of course, because computers are more powerful, we want to run 1000, 10,000. Okay, the key question is what's the percentage? Okay, not the number, not the absolute number. Is it 5,000 out of 10,000? Then it's only 50%, right? So that's something to remember. So you always need to see what the person who's presenting the, and let me tell you right now, lots of people use phylogenetic analysis, but lots of people also don't know how to interpret the results. Okay, so you have to be very careful. All right. So when we look at this tree, so let us see. I'll just go back to the presentation so that so that's clearer. So here is the bootstrap support. Some results, some papers will present results like this. Okay, this is a fully resolved tree. As Renu will remember, I always tell my students, and I always try to, until you're able to ignore those pink branches. In this case, the pink branches are not there. Just think of them as absent. Okay, until you learn to internalize and ignore those branches, it's always better to collapse those branches. Why? Because the value is less than 90%. Now, 90% support is very, very good. Okay. There was a simulation done by somebody called David Hillis and his group, in which they suggested that if you have large data sets and large trees, then their simulation suggests that something like 75% bootstrap value is pretty good. But now I see the trend that people present trees with more than 50% support. So in a sense, see, trees can be very misleading. If I want to tell you that this is my result, I can present it like this and you can believe me. If you don't know that the pink branches are really not very well supported. So if I make all my conclusions based on this well-supported tree, I may not be correct. I might say that taxon 7, say 5, 6, and 7 belong to a different genus than taxon 8 and 9. Whereas really what we know is that we are not sure of that. They are not distinct and separate. So that's how to interpret the trees. I mean, the bootstrap numbers. So a safe thing, if you have a reasonably large tree, is about 70-75% is probably okay. If you have a small tree like this, I would not believe anything that's less than 95 or so, 90-95.
All right.
And there's just one more thing. Bayesian analysis, you'll see some things there. In general, posterior probability support is generally very high. And so people prefer to sometimes use those analyses, but there are reasons why that might be the case. So there are different kinds of trees. So you have to be very careful in knowing what those numbers are and what they are. So you have to read the figure legends very carefully, assuming the author wrote them carefully. Yeah.
Next question is from Samiksha, Kamlesh, and Kamakshi. What is the difference between cladogram and phenogram?
Shall I just go to a different presentation? I'll just go offline just now and just hunt it out. I think I should have included that. I'll just stop sharing for a moment, okay?
Yes, ma'am, no problem.
Because I don't want you to see the mess that my desk is. Mm, okay, some reason I can't find the Akanksha, I don't seem to find the...
That's all right, ma'am. We
No,
can
no, no,
post
I
you the
find my
questions.
thing. I can't find the window. I just see somebody's face here, but I can't find... Ah, okay. All right, I guess I had to click on that. All right. Why does it keep coming up to the old one? Can you see this now?
Yes, ma'am.
Okay. All right. So here, if you can see this tree, I mean, several trees. So in a general sense, a dendrogram is any branching diagram or a tree. Okay. So a cladogram is a dendrogram. So anything can be shown like a tree. You can show, so you can compare two different, 3 or 4 different communities and say, these two are more similar to each other than they are to that. But then we don't mean that they are evolved one from the other, or even that they share common ancestors, right? So a tree is a device to explain data. How you interpret is dependent on what you want to do. Okay. So a dendrogram is any branching diagram, and a cladogram is specifically a diagram that we say is an estimate of a phylogeny. So like I said, tree is not always a phylogeny. Now a phylogram is a phylogeny in which the branch lengths are drawn proportional to the amount of evolutionary change, which is the kind of tree I showed you in the case of the, the eukarya, prokarya, I mean the bacteria and archaea. Okay, a phenogram is a dendrogram that shows the similarity among the terminal taxa. It's not necessarily, it's not a tree of evolutionary relationships. It is showing overall similarities. And as we saw earlier, unless, and this is the condition, okay, unless the rate of evolution is equal in all the branches, you're going to be misled. That's what happened, what my example showed you happened in the case of the eukarya relationship with archaea, right? Because the eukarya have evolved so fast according to that data, that's not always true. You cannot say that's a universal truth. So therefore, because it's so different, overall similarity wants to put bacteria and prokarya together. All right. So a phenogram is one that summarizes overall similarities. Whether you interpret it as showing evolutionary relationships is kind of up to you, but I think it's a mistake to do that, because very often it may give you the wrong. So that's why I've shown it in like that here in this figure, where I've shown that B and C are more close. In this case, you can think of these A as bacteria, B as archaea, and C as eukarya. And I've shown that B and C are the closest relatives according to evolutionary systematics or phylogenetic systematics, but phenetic analysis suggests that A and B are most similar to each other. Okay. So whether you want to interpret it as being most closely related is the step that we are trying to get away from in phylogenetic systematics.
Yes,
Okay.
ma'am. Next question is from Ramsha Khan and Shivali Sharma. And they ask, with respect to phylogenetic tree, how do you justify the precision of this statement that we humans did not descend from apes, rather we are apes?
Because we are part of that group, phylogenetic group that we call apes. We are in that, we are nested within that group that are called apes. So if you don't want to include humans as phylogenetic, as apes, then you should not, if you want to recognize only monophyletic groups, which is the purpose of phylogenetic systematics, you can't, have apes exclude human beings. That's the sense in which we did not descend from apes, but we are the group. We are part of the group called apes.
All right. This question is from Shreyasi, and she asks, which one would gain priority in phylogenetic systematics in case where at some point morphological and molecular trees contradict each other?
So there, like I said, it's pretty much open. In the sense of what people will do might depend upon your current inclinations. Many people prefer, as I said, to use the molecular data. And I'm not saying that all taxonomic endeavor will be based on molecular data, because you need the morphological data to recognize, to get diagnostic features, all of that, right? So one very nice example of this is by a paper on viburnum, which may be a little too much, but you should look at that as a kind of an example of how it could be done, which is that you build your tree based on molecular data, and then you're interpreting your characters. I didn't talk about character evolution and so on because that was not my brief, but you can interpret character evolution on such a tree, and then you can come up with where did this trait evolve, okay? And you're mapping on the traits onto the tree and saying that, oh, look, like I told you for megaphylls in the case of the land plants, right? I mean, the case of the vascular plants. We can come after the fact and say that the ferns and seed plants, ferns, by the way, include equisetum and xylotum and everything. That lineage and seed plants being sisters, those leaves that they all have, the megaphyll, is a derived state that characterizes that group. So they're being called megaphylophytes. You can give them a name based on that trait, right? So now archegonia. We want to use the term archegoniety, which is not a taxonomic or a phylogenetically valid term, because archegonia are present in bryophytes, they're present in several of the pteridophytes, right? But not in the, but not in some of the other, I mean, and so they don't form a monophyletic group. So in what sense is the archegonia? It's a convenient thing. Just like we continue to use the term algae, because it is convenient to think of this group, because they have certain traits in common, they sort of grow in water, they have different kinds, certain kinds of ecology. So in an ecological sense, it does make sense to talk about algae, but it's not. It's a kind of you don't want to then interpret that as a statement that they form an evolutionary group. So here's the point, right? Phylogenetic systematics wants the results of its activity to reflect an evolutionary system of classification and naming and classification. So if we know that a group is polyphyletic, that's why if you'll see plants, I put in quotations. Let me share again. Am I losing this thing of being able to share it? Okay, I don't know what's happened. Okay, so if you put within quotes, that is the phylogenetic systematics way of saying that here is a group that you might like to think of as a group, but remember it's not monophyletic. Okay, in the same way, apes, if you want to exclude humans, is not a monophyletic group. But you have to then think that, so you have to think about what is excluded, and you have to remember that it's the humans, because we think we are special and we want to have a separate category. So we don't want to put ourselves with the apes. So plants, algae are all not monophyletic groups. Now, nowadays, when people say plants, they usually mean green plants. Or sometimes they mean land plants. I don't know. So you have to be very specific when you're saying something. Do you mean green plants? Do you mean land plants? Or do you mean this huge group that used to include, that includes algae and is hugely polyphyletic?
Yes, ma'am. Akanksha Jha wants to know, with many data like discrete, quantitative, sequence, etc., how can we classify which one is the best?
Wait, many data, what was the first part of the question?
Discrete, with many data like discrete, quantitative, sequence, etc., how can we classify the best one?
Okay, so all data are data. in general, when you use, if you think that molecular data, so let me tell you that this is a growing field. Okay, so it's say since the 60s, but really since the 70s that it's taken on the life it has, because that's when computers became a big part of this analysis. I mean, earlier it was all manually done. What Hennig did and a whole lot of people did until the 70s was manual analysis. And it's only with computers and the sort of coming together of population genetic stuff by Sokal, Sokal on the one hand and population genetics on the other, that this kind of, analysis has happened. Okay. Now, when you look at analysis, methods of analysis, there are actually, you have discrete data and you have continuous data. And you have, you have distance data. And now you also have continuous data if you use morphometric analysis. There are ways to analyze each kind of data. Now, distance data can be obtained from both continuous characters and from just discrete characters. Okay, so methods like maximum parsimony, I'm trying to again look for that one scheme I have. My methods like maximum parsimony actually use the discrete data. So does maximum likelihood. So does Bayesian analysis. Okay, whereas things like this distance methods basically use the distance matrix, and they use UPGMA will use overall similarities. Neighbor joining methods will use a different kind of similarity. It's an evolutionary method and so on. So I'm just going to put up, if I can find the way to again to, oh, I'm sharing the screen now. How interesting. I didn't realize that. Okay. So if we look at this, okay, so I have this input data, maybe distance data, it may be discrete data or not, okay. Now, there I'm saying that the data, the method, so there's a way of kind of thinking about the different methods of analysis. You may have the method as applied on distance data, you will have what are called clustering algorithms. I've put UPGMA there, but I've graded it out and I starred it because I don't want people to use it for phylogenetic analysis, right? But you can use it for other things, like I said, overall similarities of communities and things like that. So neighbor joining method is a clustering algorithm. It uses distance data. But where do those distance data come from? It comes from molecular sequence data. So the discrete data are converted into pairwise distance matrices and then subjected to analysis. Now, I have used this frequently when you want to have a quick analysis of your data. If you want to get an idea of a very quick, very quickly get an idea of what the data are saying, this is a good method to use, and it's pretty good. It works pretty well. But again, you have to know about what models to use, this, that, and the other. We're not going into all of those. Okay, so that's the clustering algorithm, meaning it tells you, you do this, then you do this, then you do this, then you do that. And at the end, you'll have a tree. You'll have one tree. Now, the optimality criterion is one that we looked at, maximum parsimony method. It uses discrete data, as you saw. It is not model-based, which is why a lot of people don't like it. But it could give you multiple trees. They could be multiple maximum parsimony trees. So out of 100 trees, there may be 3 that have the same length. So what do you do then? Then you have to have a consensus, okay? And I haven't talked about that, but you'll come back to a polytomous tree in such a context. It's the same as the case for maximum likelihood and maximum posterior probability methods or Bayesian methods, which also use discrete data, sequence data, but they are model-based. So they're able to use the models of molecular evolution in seeking the best, the most likely, or the most maximum posterior probability of getting this particular tree. Remember, it's all a question of evaluating your hypothesis, which is the tree, based on your data. What does the data support? So these are different ways of assessing that. And a distance-based method, which uses molecules, uses models, is what's called minimum. It's a distance-based but optimal criteria method. It uses minimum evolution. Okay, so these are different ways of doing it. Like I said, there are people who are very principled. They'll say, no, this is the only correct method and this is the one I'm going to use. I tend to use multiple methods and say that, okay, if they all agree, then it's probably okay. And if not, then, either you have to delve deeper into it and then see why they don't agree with each other. That can uncover interesting patterns, but you can't make an absolute statement. Sometimes even UPGMA will give you accurate results if the rates of evolution are not different in the different lineages or different characters. Yeah. Okay.
All right. I think my next question in the list has also been answered. This question was from Ishita Chandra, and she wanted you to elaborate on maximum parsimony method.
You mean to do it again?
Ma'am, you just explained a bit. I guess that's enough.
Okay. Is that enough? So basically maximum, okay, so let me just tell you that there are huge numbers of trees possible with any number, with any set of taxa, okay? So maybe I have that nice figure somewhere to give you a, how the numbers grow, the more the number of taxa there are. So what we are trying to do is to choose the best tree among all of those trees. So the maximum, let's just look at this. Okay, so in the case of maximum parsimony tree, this is what we are asking. Here is a set of 4 taxa. To begin with, we just know that they are together. When you analyze a set of taxa, you're kind of assuming they belong together. Okay, if you're not right in that assumption, then you have to add more taxa to it. I haven't talked about outgroup, CO, and the other, right? Okay, so these taxa, there are 3 unrooted topologies possible. I haven't talked about unrooted trees, but here it is. So here is A and B are sisters, C and D are sisters. Here's A and D are sisters, C and B are sisters. A and C and B and D. Now, when you root these things, you can root them in 5 different ways. From root it here, root it here, root it here, root it here, root it here. Okay, so what are we trying to do? We are trying to use the maximum parsimony criterion to decide which one is the shortest. Okay, so that's all it is. So that's the optimality criterion. So in the case of the maximum likelihood analysis, you're trying to ask which of these trees is the most likely. In the Bayesian analysis, you're trying to ask which has the maximum posterior probability. So there are different ways of assessing that. We're not gone into any of that, but this is basically at heart, this is what we are doing when you do this kind of analysis. Okay.
Next question is from Soumya Yadav, and it says, in today's modern era, the classification is more shifted towards molecular basis with hiding of the morphological approach. What are your views on that?
My view on that is it depends on what you want to do. If your interest is in getting the best estimate. So here's another word. I've used the word reconstruction, but many people would object to the use of that. If it's a statistical thing, it's an estimate. Okay, so you can estimate the phylogeny. It's not like we are concretely sure that this is what happened in the past and you're getting that in your hand, right? You are trying to, with your data, you're trying to estimate what might have been in the past. So that's one thing. Now, when you use different methods, to use different data to estimate these things, then I always say that kicking and screaming was how I went into molecular analysis, because I'm inclined toward looking at morphology. But it becomes ridiculous to ignore data when it's there and it's telling you things, right? So it depends on your question. What do you want to do? If you're interested in knowing, okay, here's a group of plants, I want to know how this particular trait evolved, then you have to know about the trait. How does this development, is it the same in this species and that species? How did it evolve? Then you're talking the story, right? You're talking the story of these species. You want to tell the story, which is that this is what happened first, then this happened, this might have happened, this might have happened, this might have happened. So that's what it is. It depends on what you want to do with your data, what your question is. I didn't spend a lot of time doing that again because I thought I didn't have time. But if you read that Botanica article that I had sent I think to give you a little feeling for what I'm trying to say, that every project, I mean, a scientific project starts with a question, right? It's not just that you want to do phylogenetic analysis. You can't just do molecular biology. You have to have a question. When you're a student, an undergraduate student, yeah, okay, you're learning the methods, of course, but in any case, you have to understand that the basis of it is we are trying to answer some kind of a question. So, that's what it is. I don't know if that works. I mean, like here, what do you want to do with your molecular data? Right? You might want to know, what is it? I can't identify it. What is it? What is it more likely to be? Most likely to be? I can't identify it. So here will be the question. I mean, so there's a matter of, it is the case that now people talk of in the field, you can get this thing and you can do it, then you identify it right in the field. So that means very often you don't have the ability to identify it because maybe there's a scrap of it's not it's vegetative, it's not reproducing, whatever the reasons, right? But if you went multiple times to the field, probably you'll be able to identify it by the morphology. So it depends on who you are, what you're doing. You're an ecologist who just wants to do a, even if you're, you just want to have an idea of what the vegetation is, right? You may not be so interested in knowing exactly what it is or trying to spend a lot of time because you're not a taxonomist. You can't spend a lot of time identifying it in the field. Then maybe this might actually be the easier way. That seems strange, right? That you sequence it and you identify that way. We do live in strange world now, so strange times.
All right. Due to scarcity of time, I think this is the last question that we would take up here. But we assure our audience that your questions, further queries would be listed and sent to ma'am, and they will be dealt with soon. So the last question is from Muskan Khanna. Could you please mention the site or the website where we can exercise phylogenetic trees?
Yeah. What do you mean by exercise? So if you want to build, that's one thing, or do you want to exercise reading trees, interpreting trees, etc.? I'll have to kind of I don't think to come prepared with that, but I think I have sent you a link in which there are some exercises in my website.
Yes,
so you can start there, and actually from there, if you go, you can explore that, there are other sites. Like I would say that the Berkeley website is excellent for starting this thing of it, but there are really, as far as I can remember, not many places where you can just go and learn how to do the analysis on a sort of workshop type basis. So you might have to attend a workshop. But I'll look again. I mean, haven't looked in the recent past. So to introductory, there are many such sites. So just go there and look at it. And if I come across something else, I will let you know. Or if somebody else, then please email me and I'm happy to share it with others.
All right. Thank you, ma'am, for patiently attending to all the queries. Now I invite Dr. Neha Singh to present a vote of thanks to Dr. Geeta on behalf of all of us. Thank you.
Thank you.
Thank you, Dr. Ma'am. I'm feeling so privileged.
I can't hear you.
You're not audible, Dr. Neha.
Hello. Now
Yeah.
am I audible?
Yeah.
Hello.
Yeah.
Thank you, Akansha ma'am. And I'm feeling so privileged to propose a vote of thanks on today's session on behalf of Botany Department, Gargi College, and on my own behalf, I extend a very hearty vote of thanks to our dear Honorable Professor Argita. Thank you so much, ma'am, for your very, very wonderful session and insightful presentation. You made a very excellent presentation, and over it, the way you delivered it is very, very interesting and very mesmerizing.
Wow.
And
Okay.
and in fact, ma'am, this is very important. To build a building, base must be very strong. So similarly, to learn phylogenetics and to build over it, you made every concept so clear that it's just a wow. And even I would like to say that the way you have asked the question in between the session, it made things even more easier to understand. And I would also like to say thanks from all the participants that because they are also very happy and they are extremely saying that thank you so much, ma'am, for delivering such an informative, interactive, and very interesting lecture on phylogenetics and molecular phylogenetics, systematics, and trees.
Okay,
Thank,
thank you very much. That was very
thank
kind,
you
and
so,
I have
thank
to
you
thank
so
you
much,
for calling
ma'am.
me. Yeah, yeah. No, I'm happy to do this.
Yes, ma'am. Even we are blessed to have you with us. We all know